Thank you, Matthias. Thanks, Chiara. It's a great pleasure to present to you some exciting developments that we are doing together with Circle and other collaborators using the UK Biobank platform. A lot of you will remember this slide from Bob Balaban at SCMR in the opening plenary session, where he said, drawing circles, this has to stop. So this is the motivation for this talk. And I think, hopefully, I can convince you that we may have an answer to this, or starting to see answers. So what is UK Biobank? It's an imaging population. So it's basically 500,000 people in UK Biobank. It's a population cohort study, and we are aiming to recruit 100,000 from this original 500,000 people population back to do cardiac MR scanning. Of those, we will do cardiac MR imaging, we'll do brain imaging, abdominal MRI, and we'll do DEXA, 3D carotid ultrasound, and a 12-lead ECG. Because it's a population-based study and because of the scale, we're not giving contrast, we're not doing any stress studies. And we can do this at very low cost because you'll see this is not done in a hospital setting. If you're interested in the details of the UK Biobank CMR protocol, it's been published in JCMR. But just to give you a very quick overview, essentially we have 3 long-axis cines, a short-axis cine stack, 3 short-axis tagged slices with a grid, a transverse slice cine to allow calculation of aortic distensibility, flow near the aortic valve, and a short-axis mid-ventricular stack slice of native T1 mapping. So currently there's a single site running in Cheadle, Manchester. Cost about £10 million to run this so far, but since 2014, May 2014, we've scanned 15,150 participants. 2 more sites are about to open. The first one in Newcastle opening is imminent, and in Reading more towards the end of this year. Once all sites are up and running, we expect to scan 6,000 participants per year per site, so 18,000 per year, which our target is to finish the 100,000 people by 2022. This is where the first scanning center is. It's in the middle of an industrial estate, amongst lots of blood samples from people, urine samples, etc. So it's not a hospital setting, it's a very, very different setting. There's no doctor around, it's just radiographers acquiring the images. Thanks to the British Heart Foundation, we have a project grant, and that funded 2 core labs, one run in Oxford by Stefan Pichnik, Stefan Neubauer, one in our center, that have the task of analyzing 5,000 cardiac MR scans in depth manually as a reference standard. We've made good progress. So these are samples from last month. We've analyzed completely the left ventricle, the right ventricle, the left and right atrium for volumes, mass, etc. You can see that there are lots of people involved in this. So we've analyzed essentially the aortic distensibility and native T1 mapping, but there's ongoing quality control. And then we've made good progress with tagging and tissue tracking as well. Aim to complete all of this by June next year. This is what we did with CVI-42, drawing contours, endocardial, epicardial in the LV and RV in 5,065 people. We've published from these 5,065 people in 800 people roughly reference ranges from Caucasians in JCMR, again available open source. Now, we can't really analyze 100,000 cardiac MR scans, so we need other solutions. And I'm very grateful that Circle have taken it on as a challenge to them also to help us tackle this problem, and I'll show you some examples. But generally in computer science, challenges are a way of getting people with different skills to compete to try and find a good solution using machine learning. And Andrea Rai and Michael Hansen last year led a challenge, a segmentation challenge for the LV endocardial contour based on 1,000 cardiac MR scans from clinical practice. And they had very, very good experience and results, and there were very good solutions for automatic segmentation. Now, we've set something similar up within UK Biobank. We've set up a large consortium of now over 118 collaborators, over 30 sites, from 11 countries, multidisciplinary expertise, and we've got academic and commercial partners, in particular Circle. This consortium has also led to a review of the heart chamber segmentation tools for structural and functional analysis using CMR imaging. And I would like to present to you now, really hot off the press, some findings, some results from the Circle work using our 5,000 cardiac MR scans from UK Biobank that were manually segmented. So the team used a convolutional neural network for segmentation, used a training data set of these 5,000 of just under 4,000 for training the data set, and then a testing data set that was just under 1,000. And this is one example of short axis moving with endocardial epicardial contours in the LV and in the RV. And I hope you agree that this very much nicely tracks the contours. But does this really work in all of the 1,000 in the testing data set? These are results comparing the CNN, so the convolutional neural network, with manual results for end diastolic volume in the left ventricle, end systolic volume, stroke volume, and ejection fraction. You can see very, very low bias, mean difference, and quite tight standard deviations of this difference. What you can see that the ICC, intra-class correlation coefficient, between the computer and the human is very high, which is excellent. And if you compare that to what we published in our normal reference ranges paper, for human inter-observer variability, you can see that the numbers are higher, so better compared to the humans. These are Bland and Altman plots, basically showing the same thing again. Hardly any systematic bias, pretty good agreement levels, bands for end diastolic and end systolic volume. Now, some of you may know that the scanner, the Siemens scanner, can produce automatic numbers for end diastolic volume and end systolic volume based on inline tools. This is not neural network, but essentially they were also very good. Once you remove failures, there's hardly any bias for end diastolic volume and end systolic volume, these black dots. But the band of agreement is wider than for the newly trained neural network. How about the right ventricle? Again, you can see that there's pretty good agreement for RV end diastolic volume, end systolic volume, and ejection fraction. The differences appear slightly larger maybe than for the left ventricle, but again, very, very good, with very, very high intra-class correlation coefficients, which again are higher than the ones we published for human inter-observer variability. And again, the Bland and Altman plots showing you the results for RV end diastolic volume and end systolic volume, with very good agreement and hardly any bias. So what do I think the impact may be? If we have automatic segmentation, that will reduce costs for UK Biobank for sure. It will reduce delays to do research with the UK Biobank data, with the cardiac MR data, and thus it will reduce delays to translate research into patient benefit. And I'm sure you all agree who do cardiac MR clinically that it will also translate into clinical benefits for patients and reduce costs to hospitals. So we have a team that adhered to a very, very clear standard operating procedure to analyze the left ventricle and the right ventricle, and I've typed down the numbers, how long it takes, total 19 minutes to actually load, view the studies, analyze the contours really carefully. If you do this with a computer, it'll only be seconds to do this. So in summary, UK Biobank CMR consortium partners, which include Circle and others, making quick progress to tackle analysis of 100,000 cardiac MR scans. For the LV and RV, what I've shown you now, but the next is to look at the left atrium and the right atrium. And similar progress is expected for remaining image-derived phenotypes once we complete the manual analysis. And then we are already thinking about clinical CMR data sets, again, dividing them into training, validation, and testing. And I hope that you share with me, having seen some of these results, that there is optimism that we can really think about drawing circles can stop in the near future. With that, I'd like to thank the funders. I'd like to thank the Circle team who've worked hard to produce these numbers in the last month. And I'd like to thank the Oxford and the London Core Labs, the UK Biobank team, the UK Biobank Imaging Enhancement Group, and the entire Imaging Cardiac MR Consortium. Thank you.